There are many ways of drug delivery, but the most common route is the oral administration. It is easy to use and allows you to dose accurately. But have you ever wondered what happens to a tablet after you swallowed it? When a drug is taken orally, it is swallowed, goes down the esophagus, and then the dosage formulation reaches the stomach. Here we have an acidic environment with a low pH. Gastric juices, including HCl, pepsine, and lipases, are secreted, and muscular contractions enhance the digestion process. Some tablets already start to dissolve and release the active substance. In the small intestine, 90% of digestion and food absorption take place. Also, most of the drugs are getting absorbed there. Intestinal and pancreatic juices with a higher pH compared to the gastric fluids are secreted. Also, enzymes help to digest proteins, carbohydrates, and fats. The small intestine can be divided into three parts: the duodenum, jejunum, and ileum. In the large intestine, water from undigestible food gets absorbed and remaining wastes are excreted. Very important for the absorption of drugs is that the substance dissolves into small particles first, otherwise it can't enter the epithelial cells and will not reach the bloodstream. Nowadays, new methods like high-throughput screening and basic research are making it possible to find active substances with complex structures more and more quickly. So the number of new active compounds is increasing rapidly. Simultaneously, many of these substances have a low solubility, and therefore they cannot dissolve in the gastrointestinal tract. That leads to the fact that they cannot be absorbed by the body and are ultimately ineffective. What can we do about it? A common and effective method of increasing solubility in the gastric fluids is the salt formation of drugs. You can imagine it like this. The rectangles represent the free form. It dissolves only slightly in the gastric intestinal fluids. The active ingredient can therefore only be absorbed by the body in small quantities or not at all. However, with a small change in the composition, the salt can be formed. This has a much higher solubility, and the active ingredient can be absorbed by the body. Sounds great, so where's the problem? Well, sometimes something happens that is called precipitation. Precipitation can occur when the dissolved form returns to a poorly soluble free form. So what exactly is happening? Imagine an experiment where we first add the salt form to a liquid. After a while we can no longer see the salt because it has been completely dissolved in the liquid. We let some time pass and suddenly we can see an undissolved substance. It is the low soluble free form of the drug, the precipitate. The InPharma project is a fully integrated animal-free end-to-end modeling approach to oral drug product development. 13 ESRs are working on the research and development of computational pharmaceutics, biorelevant in vitro tests, and PBPK modeling with the aim of a modeling approach to the formulation of poorly soluble drugs for oral administration. Hi, my name is Laurin and I am ESR8 of the InPharma project. I am concerned with the dissolution behavior of the salts of pharmaceutical active ingredients in the human body. I am exploring the dissolution and precipitation of substances during the next 3 years. Basically, I want to achieve the prediction of salt performance in the human GI tract using in silico tools. In order to understand what exactly happens during the dissolution process, I need to collect as much and as accurate data as possible. Various experiments with different instruments are forming the basis for this. For the creation of the in silico model, dissolution data observed from the shaking flask and rotating disc are necessary. In vitro tests like USP2 apparatus or TIM using biorelevant media and conditions are simulating the environment in the human gastrointestinal tract. For the analysis of the structure of different substances, I use solid form NMR and powder X-ray diffraction. Data observed from basic experiments is used to create an initial in silico model. The dissolution of salts can then be predicted. However, the model is improved and adapted with the help of in vitro tests in environments that simulate the human GI tract. Once a suitable model has been created, the behavior of a drug in the human body can be predicted. Thank you for your attention.